Okay, everybody, we are live. Welcome back to week 1, day 5, and our feedback and Q&A for the day. So I hope that you guys are feeling ready to go into the weekend and do some professional development and engagement with some of the open science, and we've got some philosophy for you if you're not totally exhausted. We know you're totally exhausted though, but that's okay. This is like drinking from a fire hose, and all you got to do is just sit back and absorb and enjoy. So I have with me tonight, you guys all know who I am, but again, Megan Peters, hi, from California. And we have with us tonight 3 scientists from all over the world. So I'll let them introduce themselves briefly. So Rishi, can you go first?
Hi, everyone. My name is Rishidev Chowdhury, or Rishi. I'm an assistant professor at UC Davis in Northern California, and I'm split between the neuroscience and math departments. And so my research is directed at developing mathematical tools and theories to help understand how very large networks of neurons can collectively do interesting computations. You know, what mathematical principles explain how they organize information, what algorithms they run, how they effectively communicate with each other, and so on. And I do this both in kind of the abstract math world, where I try and build abstract theories. And I also work closely with data to develop new tools to analyze data, and then try and use them to test theories in data. And so I'm excited to take your questions.
Great, thank you. And Thomas, could you tell us a little bit about you?
Sure. My name is Thomas Carlson. I'm a computational neuroscientist at the University of Sydney. Our lab studies human object recognition. We use a lot of computational tools to analyze human neuroimaging data, fMRI, MEG, EEG. But we've also worked on single unit data via collaborations. And I guess with regards to dimensionality reduction, I did at one point write a philosophical paper about this, just discussing the sort of potential pitfalls and interpretation of using dimensionality reduction and sort of how we might overinterpret their results.
Great, thank you. So I'm sure that some of the bigger philosophical questions you'll be keen to answer. That'll be fun. Okay.
Philosopher. I collaborated with philosophers.
I do too. There's absolutely nothing wrong with philosophy. In fact, that's why we're doing it this weekend. I think that philosophy is really important for science. And so thinking about what dimensionality reduction buys us and what it doesn't buy us is kind of one of the points of what we're doing here. So great. And Xiqiang, will you go next?
Sure. Hi. My name is Xiqiang Yen. I'm at the National University of Singapore. I sort of started off my PhD doing modeling. So I was building models of primary visual cortex, but then got excited about doing experiments. So went to actually did a postdoc recording from the visual cortex of anesthetized cats as well as awake monkeys. And now I run a lab where we look at population coding, looking at the prefrontal cortex in working memory, and also in the primate hippocampus to look at, sort of formation of memories and recall of memories.
Cool, thanks. Okay, so I think this is going to be a really fun discussion, and we've already got some great questions. But before we get to the exciting questions, 2 things. One, there's a quick poll just to kind of get you engaged in we've only got 10 people who've done the poll. Come on, guys. There's got to be more than 10 of you here. So please do the poll. If you've seen dimensionality reduction before, it might be interesting to think about what you learned today versus what you already knew. And so this kind of leads us into our reflections, which are really important. And so actually, I'm going to kick off one of you here so I can show a slide. So I'm picking Thomas. I'm going to kick you off for 2 seconds. I'll bring you back, okay? Sorry, I only have 4 screens at once, so I have to boot somebody to show some slides. I'll bring you right back. And so what I want to show you guys is we've got this fun leaderboard here for you to do your reflections, so we can see which time zones and which pods are filling in their reflections the most. So here's our links, and they're also in that call to action down at the bottom, that blue button at the bottom of your screen. Click on that, please. If you're an interactive student, that'll take you to your reflection surveys. But this is our leaderboard from yesterday. And time zone 2, especially Authentic Jackdaw, Solid Jackal, Faithful Millipede. Faithful Millipedes are kind of killing it, guys. So maybe you want to get in there and see if you can get your pod up to the top of the leaderboard. So please do this. This is really important. Think about what you've done before, what you haven't done before, and how what we did today relates to the questions that you've been asking previously and your previous experiences and knowledge about dimensionality reduction in general. So we're going to sit here for just a couple minutes, and we're going to be quiet and try to let you go do your reflection. So please do this. I'm going to put us back up on the screen, but that call to action button is going to stay at the bottom of your screen. So please go do this now. Okay. All right. So while we're waiting, I will bring Thomas back. Sorry about that. All right, you were nearest to my mouse. So please go do the reflections, guys. And while they're doing, while all you are doing your reflections, we're going to take a look at the questions and see where we want to start. Oh, these are good questions. These are great questions today. I'm excited about this. This is going to be fun. Okay. All right. So we can wrap up the reflections now, but if you want a little bit more time, that's fine. Go ahead and do those. Those are really important. But what we can do now is we can start. Let's see. Oh, we've got 20 people of the 31 who answered the survey. 32 now, who have done dimensionality reduction. That's great. I'm wondering how many of you guys did like type PCA into Python or MATLAB or something versus out like we did today. So maybe we can talk about that too, what you learned there. So cool. So let's do some of the questions then. We can see where we want to start. So the top ranked question here comes from Yasuo Kabe. And this question is, what specific dimensionality reduction techniques would you choose for answering different questions? For example, PCA, T-SNE, UMAP, etc. So maybe a good way to do this one would be to talk about how we all have done dimensionality reduction and used dimensionality reduction in our own research. So who wants to go first?
I can jump in just quickly. So I used PCA for a long time, but primarily just to sort of reduce the amount of data. So we'll use classifiers, and by sort of narrowing the amount of data you feed the classifier, you can get better performance and faster performance. So PCA is just a simple way to take a big piece of data, make it a little bit more smaller and manageable for the classifier. We've also used SNE more recently, more for just visualization. So part of our work is we'll try to reconstruct representations in the brain. We call them representational spaces. So SNE is a nice way to sort of get a visualization of like how different objects relate to one another in a particular brain representation.
And when you use PCA, do you primarily use it like just plain vanilla linear PCA, or do you do any kind of the non-linear fancier versions?
Yeah, it's strictly linear just because it's the only goal is to reduce the number of dimensions. So when you do EEG and MEG, there's a whole lot of correlation between the channels. So you don't need to be feeding in 128 electrodes or 64 electrodes. Because often you can capture 98, 99% of the variance with half that, or sometimes even third or even less.
Cool.
Yeah, I think we have used PCA also to try to remove some noise from the data. So sometimes when we look at neuronal recordings and we're trying to do full trial analysis, that gets very noisy. So sometimes we do that where using PCA, you can kind of remove some of the lower components to try to remove some noise. So that's also sometimes useful to do.
And so I guess for me, so here are a few, I think, good, useful general principles to think about. The first one is you should start with simpler methods and then move to more complex methods. Simple interpretable methods, if they work, then what the structure you're seeing is probably real because you haven't put some big fancy complex thing in there. And often the structure you see is easy to make sense of. So simple is better than complex, but start simple with something like PCA and then build up. A second nice principle is that if structure is real, if it's really there, it's often visible using multiple methods. Some methods might show it to you better than others, but if you're able to see the same structure, using PCA and IsoMap and t-SNE, that's great. It means it's probably there. The third principle, again, and this is kind of a Bayesian principle, is to think about what you know about your data. Certain kinds of methods work well for certain types of data. For example, if you think the trajectories are smooth, if you think neural trajectories are smooth, your firing rate doesn't suddenly jump from 20 to 100, then there are all these methods like Gaussian process factor analysis that really build that smoothness in. And so if you can make assumptions, it's great because you get more. Of course, you've made those assumptions. And if those assumptions are wrong, it can mislead you. Kind of for practical matters, PCA is great to start with. Things like factor analysis have noise models, which can be helpful for getting rid of extra noise. t-SNE and UMAP, often they're great for visualization. They can be complicated to interpret for more general dimensionality reduction. And then there's a whole suite of these manifold nonlinear methods that when they work, can work really well, but they sort of fail a lot. They can be kind of brittle. You change certain assumptions, things go wrong. The nice thing with all of these packages is you can just have a workflow where you start with something simple and just step through a whole bunch of things and then just look and try and make sense of why one is failing and one is not. But yeah, both principles start simple.
I tend to use them, the simple ones as well, to the extent that we do this, just kind of for the same reasons that Thomas was talking about, is that in human neuroimaging data, you have this paucity of data. You never have enough data. And you always have way too many dimensions when it's like fMRI. Maybe you have all of these dimensions and you have to find a way to do kind of something before you do any sort of decoding or any sort of other kind of representational analysis. It's nice to do the very simple thing to see whether there is anything resembling your expected structure in your data. It gives you a sense of whether you're looking in the right spot, whether your data are clean, whether you've kind of done your pre-processing right almost. So that tends to be how on the basic side we use dimensionality reduction like PCA in my lab. We also do some kind of non-parametric or parameter-free dimensionality reduction with autoencoders, and then looking at the latent variable like that bottleneck space in autoencoders. But we won't get to autoencoders until much later in the course, but kind of keep in mind that this is one way that you can actually still also use autoencoders, is to do dimensionality reduction, but without some of these constraints like linearity that are kind of baked into some of these other things. Cool. Okay, so I hope that answers your question, Yasuo, about how maybe these different ways of doing dimensionality reduction can be applicable under different conditions. The short answer is kind of try some stuff, but then also, like, depending on how correlated your features are going to be or how much independence you can assume your features to have, there might be some that work better than others. If you really want beautiful visualizations, t-SNE is kind of fun. If you really want, to get at the very basic structure of your data, then linear PCA might be exactly the tool that you need. Cool.
Yeah, but I think Rishi's advice was very good. I think, starting with something simple, I think is always sort of the best strategy, because I think with some of the more complicated methods, it gets harder to and harder to interpret. So, start with something simple, and then, if it doesn't kind of get you what you want, then maybe try something a little bit more complicated, right?
Yeah, that's something that we've really been emphasizing actually is think about what you want to get out of your data set and out of your analysis and out of your model. And that's going to dictate which model you pick. And so if your goal is simplicity and interpretability, don't build the crazy fancy nonlinear thing first. Start with the simple thing and see what you can learn from that will maybe help you build a fancier, more complex model with more parameters later, or something at a different kind of level of analysis. But simpler is better. We've talked about Occam's razor. We've talked about modeling goals, that kind of thing, all the way through the course. So, very good advice. Awesome. Thanks. Cool. So so I hope that answers your question a little bit. Gives you a good kind of broad overview of the types of ways that we can use these different things. So we have another question now from Joe Goodsell saying, could someone comment on the pitfalls of dimensionality reduction? And specifically, what to look for when assessing whether a result might be trivial or misleading? Good question. I like these questions because it's like, how do I know that I did it right? But also, how do I know that what I'm looking at is not just the result of a black box fancy math thing that, like it's actually meaningful? So anybody have, anyone want to jump in here?
I can kind of give you a couple of thoughts. I think one thing is it helps to know what your dimensionality reduction method is trying to do. So for example, PCA is trying to account for variance. That's great in some settings, but imagine you have one neuron, a voxel, that for whatever reason is just firing really high. Your PCA is going to really, really care about that. Maybe that's a good thing to do. Maybe that actually means there's a lot of signal there. But also in the brain, some neurons just fire more than others. And so maybe that's not a good thing to do. And so the first point is it helps to know your method. Two other more practical things. One is just look at the numbers you get and worry about really big numbers. Often a sign of something going wrong is when you have big numbers that cancel out, or numbers that are much bigger than the data you put in. That could be a sign that the method's kind of doing something odd. The second thing is to worry about really sparse vectors. Often what you're looking for in dimensionality reduction is some kind of shared structure, some kind of structure that applies across neurons, that applies across voxels. If one of your dimensions really cares about just one neuron, just one voxel, again, that's a sign that it's like pulling out something that maybe it shouldn't.
Yeah, I can I'll build off that a little bit. So one thing with when you're doing PCA for dimensionality reduction, often you'll set some sort of cutoff, like I want to retain 99, even 99.5% of the variance. But for human neuroimaging data, a little bit of head movement in the scanner, that's going to make a huge amount of variance, and that's going to soak it up. So you might, by setting a 99% cutoff, there might be an interesting signal that actually falls under the threshold that doesn't make it to the sort of next stage of analysis. The other thing with regards to assessing your data, and this is sort of the commentary where we got into with the philosophers, is you always have to remember that you, the scientists, are the interpreter of the data. And when we discussed in the context, I'm trying to remember the psychological phenomena, it's really hard to say, like pareidolia or pareidolia, but it basically is when people see, Jesus in a piece of toast, faces in the clouds, things like that. And the brain is really good at bringing interpretations to data. So I just, I'd sort of comment it's sort of hard to figure out when it is misleading or might be real, because your brain is superimposing its own biases onto the data, whichever method you use.
Yeah, there was a whole...
Yeah, I think the other point...
Please, go ahead.
No, sorry, go ahead. Just go ahead.
I was just going to say that there was actually... a couple of years ago now, there was a whole workshop on manifolds and dimensionality reduction, but specifically manifolds at the cosine workshops. And And I think it was even a 2-day one, if I remember correctly. I might be wrong. It felt like a 2-day one either way. And it was all about how, we're very excited about dimensionality reduction and discovering using dimensionality reduction to discover the dimensionality of the representation in the brain, especially in more higher cognitive areas where the representation is likely to be complex and where there is kind of mixed selectivity among like the neural preferences and so on. And a lot of the discussion in that workshop centered around the idea that you discover using dimensionality reduction and how many dimensions capture X% of the variance. You discover the dimensionality of the task that you are asking the agent to do. You don't discover the dimensionality of the representation in that area of the brain. If you give a rat 2 options, what you're going to find is that a dimensionality of 2 is likely to account for a large percentage of the variance in what you're looking at. So I thought that was kind of an interesting way of turning the problem on its head and saying, well, we would like to use dimensionality reduction as a tool for reducing the number of features to input into models and so on, but we also shouldn't put too much stock in the number of dimensions necessarily that come out of the dimensionality reduction, because we need to be very careful, as Thomas was just saying, that we don't go find exactly the thing that we were looking to find. That we recognize that we've set ourselves up to discover a two-dimensional representation if the animal is doing a two-dimensional task. And I'm sorry, Xicheng, I cut you off there. Did you want to add?
Oh no, yeah, no. I just would I think wanted to point out that I think this is sort of addressed during the tutorials as well. I think the dimensionality reduction techniques sometimes build in orthogonality or independence. And so sometimes you got to be careful about sort of, whether your data actually, sort of follows that, right? So, a way that the result could come up to be misleading is that, if your data is actually not, really represented well with an orthogonal basis, right? And so then now you start to think, oh, maybe there's something there, but, it's sort of a byproduct of the technique. So I think there are sort of different ways to look at these things, and you have to be very conscious of sort of what is it that you're looking at, right?
Definitely.
Yeah, I was to say that's a really good example. I think I remember the example of multiple voices in a room. If you run PCA on that, it's really bad at sort of figuring out the different speakers. But if you use a different technique like ICA, then you can pull out the speakers, and that's because the dimensions aren't orthogonal.
Yeah. So always think about whether your assumptions are right when you pick a tool. Cool. Okay, so let's go on then to the next one. So this is an interesting question. I'm not quite sure how to answer this, but maybe one of you will want to jump in. So the question is Leandro Scholz, if I'm pronouncing that correctly. The question is, with neuronal activity data, dimensionality reduction is most often used with dimensions as the number of neurons and number of observations as the number of time points. Could we talk about when it would be useful to do the opposite, use the number of time points as the number of dimensions as input and the number of neurons as output? So I don't feel like I have the right way of answering this, but maybe one of you guys want to jump in.
So I guess it's interesting to note that PCA already does this. If you think about what PCA is doing, if you remember from your tutorial, you got these weights and you got these scores. And when you throw away dimensions, if you go down to k dimensions, you're keeping k weight vectors and k score vectors. And so depending on what you call what, one set of patterns gives you certain correlation patterns across neurons. These two neurons like to go up and down together. These three neurons are anti-correlated and so on. The temporal dimensions give you time patterns that the network likes. So maybe one time pattern is things tend to go up. Another time pattern is things tend to go down and then go up. What PCA does is it multiplies these two together. It says, so maybe these two neurons like to go up, and they're coupled to the time pattern that increases, and so you'll see them both going up over time. Maybe these two neurons are coupled to this time pattern, and so you'll see this kind of like, depending on how you frame PCA, though, you basically see the structure come out very nicely. There's a formulation in terms of what's called the singular value decomposition, and that will give you both the time patterns and the neuron patterns, and you get them at the same time.
Cool. That was a very...
Yeah, I mean, I think I think it is true that I think a lot of times we try to reduce dimension, in terms of number of neurons, so that we can kind of see, what's happening in time. But yeah, I think there are sort of other ways where you can also kind of look at the trajectory, right, of the activity. And so maybe that is an aspect that maybe is not as well used, it's not used as common. But I think it is potentially also quite interesting to look at.
But by trajectory of the activity, you mean the trajectory of the entire population of, features or voxels or whatever you're looking at through this lower dimensional space?
Yeah, so it's a little bit like what kind of Rishi was talking about, where, sort of if you imagine that I have, like a very simple example would be some sort of oscillator, right? And so I could have some sort of oscillator that is sort of one dimension. And, can I layer some other sort of time varying signal on top of that, right? So can you think of sort of the population sort of as a sort of a point that's moving in time? And so, the way that we often look at it is try to say, okay, well, we're going to collapse all the neurons into one spot, and then, try to look at the trajectory as a function of time. But maybe you can actually look at it a different way, which is to look at, sort of how these neurons are changing in time, right?
I see. Yeah. Cool. All right. Okay, well, I hope, Leandro, that was satisfactory. There was a follow-up that was how would that relate to behavioral data, but I think we kind of touched on that. Well, maybe we didn't. Do you guys want to have anything to add in that regard, how it might relate to behavioral data?
There's fun work that tries to do joint neural and behavioral dimensionality reduction. And so you can do these things where you can do dimensionality reduction on behavior and say, well, what are stereotype patterns of behavior? Let's say I'm doing some crazy thing, and you're like, well, let me do dimensionality reduction, and maybe I do this a lot, and maybe I do that a lot. And then you can kind of like couple those two neurons and say, well, what low dimensional neural pattern is most correlated with that, and what other low dimensional neural pattern is correlated with that. So I know some of the folks at the University of Washington have done some of this, for example, and trying to be quantitative about behavior is kind of a hot area, I think, in neuroscience in general.
Yeah, and for motor movements, the old term, or older term for that is muscle synergies, right? This idea that you have, a low dimensional kind of signal that goes down, and that you have these kind of preconceived or pre-programmed motor movements, and so those are ones that kind of occur in a lower dimensional space due to like the constraints of biomechanics and so on. So I think maybe when we get into optimal control, we may be talking about this a little bit, because I think there's going to be some reaching and grasping in that section, but I'm not sure. Okay, cool. So why don't we move on then, and we can do the next one, which is up here at the top from Rebecca. Does the brain have a mechanism like dimensionality reduction? So I know how I would answer this question, but I will let you guys go first.
I love this one. And I didn't know if everybody agreed, but I would say absolutely. You just start at retinal ganglion cells, and they're not interested in encoding just, the individual receptors. They do center surround, and that's a form of dimensionality reduction. And then you go to primary visual cortex, and you see kind of like Gabor-like filters, and I'm trying to remember the original work. Olhausen and Field basically showed that these filters are really super efficient for encoding the natural world. There's all sorts of different worlds that could live out there, but we live in a particular world with particular statistics, And V1 neurons basically look like they're sort of optimized to encode natural images, and in a compressed way. And I think it was also Barlow talked about this, but basically one of the goals of sort of brain representation in the visual system is basically to sort of minimize, sort of compress the space, because firing neurons is really expensive. The brain, I believe, is about 20 or 30% of the body's metabolism. So I can imagine if all those neurons were firing all the time, it would probably be in the 50 to 60% range. So there's good reason for the brain to do this type of reduction. Awesome question. Love it.
Yeah, good question. Anybody else want to go?
Yeah, I would say I agree as well. I mean, I think, I think if you look at sort of how the neurons connect to each other, right? I think there's always this sort of, weightage of, some connections are a bit stronger, some connections are a bit weaker. And I think that's probably a form of sort of dimension reduction, right? It's sort of selecting out the information that it can process. So I think sort of this idea of, each stage of the processing, right, sort of capturing some information and doing some transformation, I think it's sort of probably, I think it's the correct way of thinking about it.
Well, I'll jump in. So I study metacognition and consciousness in humans. And so there's actually a lot of talk about dimensionality reduction in metacognition and in perceptual awareness and attention, that the result of attentional selection mechanisms and the result of, like, something that one of the mechanisms that pushes information, especially from perception and memory, into, for example, the global workspace, or some of these other kind of theories of consciousness and phenomenal awareness. Dimensionality reduction and compression is actually one of the mechanisms that gets talked about. And so Yoshua Bengio has a archive paper on this from actually now a few years ago now, 2017, that is called The Consciousness Prior. And it is all about dimensionality reduction and how the result of dimensionality reduction and compression is the abstraction that leads to conscious awareness and functions associated with consciousness and higher cognition. And so, like, all the way from, we were talking retinal ganglion cells, and all the way through to consciousness, which is arguably like the closest to philosophy that you can get in neuroscience. Basically all the way through this processing hierarchy, it seems that compression and dimensionality reduction is kind of the name of the game, and is a critical, or hypothesized to be a critical feature of how our brains actually process information. So there's got to be some way that the brain is actually doing it.
Yeah, and I just built on that even further, because it's interesting. Like, you think of, I mean, I'm a vision scientist, so if our brain had to process all the little retinal ganglion cells, it'd be overwhelmed all the time. So when we look out and perceive the world, what we see is objects. Well, from my perspecve, is not person object recognition, but the brain just immediately parses the world not into like gray square thing with light edges. It sees computer, and it's sort of, and then those are sort of the basic units of sort of our perception and interaction with the environments. And that's just a big, huge leap in dimensionality reduction, to go from retinal ganglion cells to individual objects in the world.
And you might even argue that something as kind of core to cognitive psychology as like geons or something might be the result of a dimensionality reduction scheme. It's like, oh, well, now I see this geon instead of this collection of edges and features and specularity and so on. So yeah. Yeah,
Biederman actually wrote about, I mean, that's why he loved geons, is because it's like, hey, it's a super compressible format for a complex world.
Yeah, exactly. Which allows us to like function, as you said, that if we were having to constantly parse lines and edges and changes in luminance as a result of the sun going behind a cloud, I think we would probably not get very far as in an evolutionary sense.
And you could make the same argument ready for the existence of space even. The brain creates space to organize the world as a way of, like compressing and organizing structure. And obviously that's an old idea. But yeah, it's really everywhere.
Great question. Really enjoyed that one. Thank you, Rebecca. Good question. Cool. Okay. So now we've got another one that's kind of a big idea question from Sophia. She's asking, do you have thoughts on interpreting the components that arise from PCA? Could you help us to understand where it might be appropriate to interpret their meaning and where it's more appropriate to just use PCA as a method of reducing the complexity of data? So this is kind of hitting on some of the things that we've been talking about here, but maybe we can go into a little bit more detail if we've got maybe some examples from our own work or something like that that might speak to this as well. What do you guys think?
So I could go back and use the examples I used of, neural patterns and time patterns. Again, just to be concrete, let's say you've got a matrix, you've recorded from 100 neurons, and those are your columns, and you've recorded a whole bunch of time points, and those are the rows. And then you do PCA, and you get some set of neural vectors that are the patterns, and some set of time patterns. And the time patterns often will tell you what the network's doing. So for example, in a working memory task, you might see a time pattern that turns on and stays there for a while and turns off. And just plotting them, you'll see that. Or if you're preparing to make a decision and you just plot the time pattern, you'll often see it ramp up towards some threshold. The neural patterns tell you which things like to go up and down together. And so if you look at if you have 100 neurons, each neural pattern will just be a 100-dimensional vector. If you make a scatter plot of that, and say if you see that entries 1 and 10 and 37 are all high and positive, and entry 21 is negative, that tells you that neurons 1 and 10 and 37 tend to go up and down together, and neuron 21, or whichever number I used, tends to be opposite to those. And so the nice thing is the PCA components are often pretty interpretable in this simple mathematical way. What it actually tells you about what the underlying system is doing is maybe one step further. But the nice thing about these linear methods, it's really just multiplication and addition. And so when you plot them, it tells you what time patterns is the system like and what neurons or voxels like to go up and down together.
I'll jump into that also from the perspective of human fMRI. So maybe not necessarily PCA, but other methods of dimensionality reduction. If you then go and look at kind of the clusters or the dimensions that arise from doing this, you might get a sense of whether you've discovered something meaningful. And just, I'll say this with the caveat that just because when you look at how your data clusters, it doesn't make sense to you, that doesn't mean that it's not clustering meaningfully. It just means that the way that the brain happens to be representing these concepts is not something that maps onto your particular conscious cognitive way that you think that the brain should be clustering things. But one of the examples that I really like of using this kind of dimensionality reduction, and I can't remember, but I think it is just plain vanilla PCA, is Jekyll and Alex Huth's work. The pragmatic data set and the pragmatic way of looking at representations in the brain. They have people listen to stories in the scanner and then look at the clusters essentially of brain patterns that went with concepts, ideas, love, family, numerosity, concrete objects, this kind of stuff. And so what's cool is that you can actually see the clusters and you can see the dimensions that come out of it. And they have this really beautiful visualization of the dimensions that go from almost positive to negative, or from concrete to abstract. So I think that looking at the clusters and the dimensions that arise from some of these dimensionality reduction techniques, it's a little bit of a just look at your data and you'll find the answer. But it's true. Sometimes it actually becomes very interpretable if you just plot it and you look at which categories and which stimuli that you looked at land where in this 3 highest PC spaces. So that would be kind of my contribution there. I think. I don't know if anybody else has something else to add.
Well,
No?
I think I would again sort of caution. So I think we've come across cases where say we have a working memory task where we have sort of a few different locations, and we try to kind of run PCA, and you find sort of one component and then an orthogonal component. But really, actually, if you kind of look at the data, it really is not orthogonal, right? And so what ends up, what you're getting is sort of, again, the component that has the largest variance and then the next one that has the largest variance. But if you kind of look at the data more carefully, you see that first of all, they're not orthogonal, right? So the PCA components there actually are not so meaningful. So I think a lot of times you just have to be very conscious about, is your data likely to be orthogonal, right? And so then can you actually interpret that component? So I think those are some things that I think we've made mistakes ourselves to kind of sort of have to kind of constantly remind ourselves that sometimes you got to be very careful about interpreting these components.
Yeah, I'd also echo that as if you know that your data follows the assumptions of PCA, then you can dive in head first with the interpretation. The problem is working with the brain is I would say very rarely does the data follow the assumptions of PCA. In which case you're sort of you have to do a little bit of searching, but there are other ways to sort of deal with these issues. And then one was like the mentioning of Huth's data. So if Huth was one of the first studies to ever study the brain, then you should walk in with all sorts of skepticism of his data. But you can see some of the mappings in his data is consistent with previous work. So that tells you that even if it is going wrong, it's not going wrong in a very big way. So you can use other data to sort of support the interpretation of the output, even though maybe you don't sort of fully satisfy the assumptions of PCA.
Yeah, this goes a lot with the kind of conceptual understanding that we've been trying to build across the sessions so far. There have been some questions in the previous Q&As where it's like, how do you know that you picked the right model, or how do you know that your model is doing something meaningful? And one of the ways to answer that question is look at what's been done before. And if what you found is at least not completely wacky out there crazy in comparison to what's been done before, then maybe you're on the right track. And if it is wacky out there crazy, then if you can find in your experimental paradigm or design why that would be the case, then you've also discovered something very interesting. But if you tried to replicate something that somebody else did and you discovered that vision actually occurs in the prefrontal cortex or Heschel's gyrus instead of in V1, then you're probably barking up the wrong tree. So yeah. Okay.
Cross-validation is good for this too. You can always cross-validate.
Yes. And we've talked about model fitting and overfitting a couple days ago, and so that's another way of doing this too, is to discover, does the number of PCs that you need to capture X% of the data, or what those PCs seem to mean, change drastically between your training set and your test set? Then if it does, then you're probably in trouble. Okay, cool. So our next question then is also from Leandro. We've got a couple people here who are very active. Other people ask questions too. That's cool. So, but keep them coming, Leandro. That's great. So there have been discussions about the use of t-SNE data for clustering, but it was developed as a visualization technique, and it is dangerous to use it to cluster data given stochasticity and the free parameters. What do you think about this? Would you use t-SNE as an input for a further clustering step or not? What do you guys think? I use t-SNE basically for visualization, so I don't think that I'm qualified to answer this particular question.
I also feel unqualified. I'm going to defer to the other guys. I'm a visualizer.
I've also only used it. Yeah, I've only used it for visualization as well. I don't know how to interpret it otherwise, so I haven't used it in other ways.
I guess I would say two things. I guess in general, clustering is kind of a brittle problem. If you change parameters, things can cluster very differently. And there are all these interesting theorems that say, there is no best clustering algorithm. And so with any clustering algorithm, you will have free parameters, and you should always change them and see how the clusters behave. That's always something you should do. But having said that, I would probably use something like k-means for clustering rather than t-SNE, because t-SNE is not trying, even after you've done t-SNE, it doesn't give you the clusters. You then have to go in and somehow say, well, this cluster. So you're anyway using something like k-means on top of that. So you might as well use k-means to start with. You often want to do a dimensionality reduction step like PCA first before you cluster, just because stuff behaves can be difficult in high dimensions. There's lots of ways for things to go wrong. And so you often use something like PCA to get to maybe 10 or 20 dimensions and then do your clustering there. But that's just a pre-processing step. But yeah, I would use k-means over t-SNE, but I would change the k, I would change some of the parameters.
Would you recommend also doing that dimensionality reduction step before you do t-SNE? That's kind of what we've been doing, but it's not a core piece of what we do in my lab. So I'm always curious to know what is a kind of, especially for, in my case, human neuroimaging data, what is a good number of dimensions to feed into t-SNE so that you can get something that is kind of reasonable and meaningful and useful on the other end? Sorry, I'm jumping in and asking my own question.
I would probably like you to talk to domain X. I feel like a lot of stuff is just like so domain specific that I'd be nervous about. I think a lot of the time it's just like, yeah, answers are very domains. But I think you should do some kind of dimensionality reduction first is generally a good idea.
I think that's really informative, though, to say that it's like there isn't an answer for how to do the dimensionality reduction before you do some sort of clustering, because it does depend. So it's helpful to me, and hopefully it's helpful to the folks who are listening too. Thanks. Cool. Okay. So it seems that the consensus is no, probably don't use t-SNE as input for a further clustering step. Okay. All right. So now we have a question from Mango. Oh, hang on. They start jumping around when we've got competing votes. So I'm going to do Mango's question first. So one question about today's tutorial is, today we learned how to do dimension reduction in sample X variable data, sample by variable data. But how do we deal with the time series data in dimensionality reduction, like a data set with trial by variable by time, such that we could see the reduced data moving on the low D plane with time? And there's a comment here about the introduction video in which Byron Yu talks about the neural trajectory in this low D space, and that follows up and talking about the interpretation of this low D trajectory. So I've got some thoughts of my own, but I will let you guys go first if you'd like. Do you guys ever look at trajectories in low D space and try to interpret them in a particular way?
I guess there are a few different ways to, this question is also actually similar to the one about tensor decomposition above. If you have data so I guess there are two questions. One is if you have variable and trial and time, what do you do? And then there's the second question about trajectories. If you have variable times trial times time, you can do one of two things. You can have your variables and you can concatenate the trials. And so you have, time 0 to 100 for the first trial, time 0 to 100 for the second trial, and so on. You could do that. Or tensor decomposition is a pretty hot area of research now. And so, for example, people from Surya Ganguly's group at Stanford have been doing some of this. Mm-hmm. So you can just Google around and find that. And then thinking about trajectories is often a great way just to see how a system is evolving over time. You can do that using a method like PCA, which doesn't impose any kind of trajectory structure. But if you're interested, you can also Google something like Gaussian process factor analysis, which tries to look for smooth structure tries to look for smooth underlying trajectories. Or you could Google LFADS, latent factor analysis. There's kind of a whole host of these methods. I think once you understand PCA, though, that's kind of like a great sort of intuition pump. Once you have that, and we really understand that, you can understand these other methods as kind of adding on assumptions to that or changing it in different ways. And so I suggest really digesting PCA, really chew on it until you thoroughly understand it, and then think about other methods from that lens.
So I'll follow on that, which is I think that it would be important to say, well, if you're going to do this kind of trajectory through low dimensional space, and you're going to try to interpret it, the first question I would probably ask you is, well, what is the animal or organism or agent or observer or whatever system you're studying actually doing right now? Why is the trajectory through space kind of interesting? Are you interested in watching like an evolving decision variable over time in response to a visual stimulus? Are you interested in looking at an evolution through something like state space in a resting state fMRI scan? And so I think that in some of those cases, the evolution through this low dimensional space could be potentially very interesting, and also the derivative of such a trajectory, like how quickly it moves through that space, would potentially be very interesting, or potentially completely boring, depending on the question that you're actually interested in. And so if you're trying to understand decision variables and how a system or a decision evolves over time in response in like a perceptual decision-making task or a motor planning and execution kind of task, then that would be potentially a really interesting thing to look at. Or it might be totally irrelevant to your goals. So yeah, that's kind of how I would go. Does anybody else have anything that they want to say?
I would just say, like, yeah, we've actually dealt with this in the practical one, and we used one of the solutions that Resh Deva suggested. Is basically we just stack the trials on top of one another, because we were doing MEG and feeding the MEG data into a classifier. So we want to reduce the number of dimensions. So you just stack the trials, and then you can sort of do the dimensionality reduction, and then you can unpack the trials back out to run the classification analysis.
But I think there's also kind of nowadays people are using like things like DMIX PCA, where I think you can kind of label some of the trials, and so that's kind of like a natural extension of PCA to kind of look at these types of things. But I think sort of this idea of the trajectory, I think Byron sort of in his lectures actually kind of gave this nice example of sort of if you have a manifold, learning within the manifold may be more sort of easy for the organism to do, but then things out of the manifold may be kind of more difficult, right? And so this kind of trajectory analysis could be kind of useful for those kinds of insights.
I love Byron's work on this. I think it's really, really beautiful and really interesting. And to look at how the animal actually can learn to use the out of manifold representations, but it like barely, and only under certain conditions.
Right.
And it speaks a lot, I think, to when you're discovering lower dimensional representations in fMRI data as well. Like in theory, each voxel contains however many thousands, millions of neurons, and so like a particular neural pattern should not be unique, or that a single voxel pattern can go with, in theory, any number of actual underlying neural patterns, but maybe not actually, because of the fact that there are these kinds of hard-coded connections that you see. There are these manifolds underneath it, and so that in that sense, that's why MVPA with voxels actually works, and why you could actually discover something with these massive 3 millimeter things that contain so many neurons, even though underneath the representation is much higher dimensional than that. Cool. Okay. So do we want to do one more? I know that we're right up at the edge of the hour. Maybe we do one more. Okay. So we'll take the top one here. Lena Liu is asking, if the data has 3 dimensions, for example, a video clip, how should we conduct dimensionality reduction? So a video clip would be, I guess, just a flat thing over time. Is there anything like, I guess this is tensor eigenvalue decomposition, or maybe using something like nested PCA where you can compute PCAs from PCAs?
I just sent out a link actually about that, because it's the last question too. But yeah, so this is like a hot area of research. Yeah, totally.
Can you give us a TLDR on that?
Yeah, so you can basically so the idea of PCA is to take simple structures and glue them together to make a more complex structure. And the simple structures are linear. And really you can think about PCA as taking your big data matrix and writing it as a bunch of simpler matrices, as by adding up a bunch of simpler matrices. Now imagine a matrix. Instead of a matrix, you have some structure with 3 dimensions, like a cube. Maybe that represents your video over time, or maybe one dimension is neurons, one dimension is times, one dimension is trials. So whereas PCA works on flat things, you can imagine a cube or higher dimensions. And then you can write that cube as a sum of simpler cubes, the same way you can write your square matrix as a sum of simpler matrices. And so it turns out a lot of the intuition that you use for PCA, you can kind of extend to sort of further and further dimensions. And again, once you understand PCA well enough, that it's not super complicated to think of how you take your squares and make them cubes, or you make them hypercubes.
Perfect, thanks. Okay, well, we're right up here on the hour, but I want to give you guys a chance, if there's any parting words of wisdom that you would like to share about dimensionality reduction, some of the questions that we talked about. I know we didn't get to everything, so before we do this, I'll say, if we didn't get to your question, take it over to Neurostars, take it to your pod, take it to chat, take it to TA, there's other errors, take it to your TAs. Just because we didn't get to your question doesn't mean it's not a good one. So please continue the conversation offline, or maybe even tomorrow, as you kind of absorb and marinate over the weekend, because I know it's been a long week. But yes, so just because we didn't get to your question doesn't mean it's not a good one. So please take those to your pods and keep the conversation going. But do you guys have any parting words of wisdom that you'd like to share about dimensionality reduction, its power and its pitfalls, and its future in your lab or in other work?
Yeah, I'd say, it's a powerful, wonderful technique, but always, you always have to, it always goes back to the assumptions of the methods that you're using. I really like the suggestion earlier that, you sort of start simple and move complex. Always, if you don't know those assumptions are in place, then, approach the interpretation with caution. Yeah, I think that's pretty much it.
And I would say something similar. You want to keep thinking. You don't want to kind of shut off your brain and just use this as a tool that you don't understand. The 2 things you should understand is understand your problem and understand your tools. Are you building a chair or are you building a table? Do you need a hammer or do you need a saw? In order to do that, you need to understand, what problem am I trying to solve, and what kind of tools do I have, and keep thinking about how those things fit together.
Yeah, so I think the one thing that we haven't sort of talked so much about is sometimes it's actually helpful to try to simulate data. Sometimes it's also difficult because, it's hard to kind of get that kind of richness. But sometimes, if you're testing something out, it might actually be useful to try to simulate it and then apply a method to it to kind of see what you get, right? So because sometimes what you see is that actually, you can create something that, this method will pick up and, it's just an artifact, right? So I think sometimes it might be useful to try to simulate your data to kind of see what is it that you're trying to get at, and can this method actually pick it up, or does this method kind of screw it up, right? Does it mix it up, inappropriately? So that could be something that is also, kind of goes hand in hand with this, these methods.
Great, thanks. Yeah, I think I don't really have too much to add. I thought the questions actually were really, really good about how to use these things appropriately. And as someone who for a very long time was really only an end user, and I still really am a little bit of an end user of some of these things, and I do tend to type PCA into Python or MATLAB and hit enter and then work with the results. But it's really important to think about what the underlying assumptions are, and also the interpretability of the outcome. And don't just say I did PCA and I found these dimensions and look, it's a beautiful Nature paper. Although some people can do that, but it's not just about that. It's about understanding whether the interpretation that you're coming to as the result of using some of these very powerful techniques is something that answers a question that you are interested in looking at. So Cool. Okay. Well, thanks all three of you. Thank you very much, Rishi and Thomas and Xicheng for being here tonight. We really appreciate your time and your expertise. And I certainly had fun. So I hope that you guys will join us back for some other live events. Tune in if there are ones that you are interested in participating in, even in the chat. And we'll see you there, I guess. Okay.
Thank you. Well, thanks,
All right. So
Thank
thanks
you.
very much. This was
Bye.
great. We'll see you guys next time. Bye.
Bye, everyone.